Have you ever really thought about the absolute limits of what we can see? I'm not just talking with our eyes, but with our most powerful microscopes. What if I told you there's a hard wall, a physical barrier, that for over a century we believed was just unbreakable? And then what if I told you that scientists have figured out a way to smash right through it, not with a better lens, but with a ridiculously clever piece of software? Just look at this. On the left, you've got this blurry, kind of hazy view of microscopic filaments. That's what a really good, top-of-the-line microscope gives you. But on the right, well, that's the exact same image after this special software has worked its magic. The difference is just staggering, right? It goes from a vague glow to these incredibly crisp, distinct threads. So that's the big question we're tackling today. How in the world is this possible? There's no new hardware here, no fancy new microscope involved. This incredible leap in clarity is achieved purely through computation. It feels like a magic trick, but it's absolutely real, and it's completely changing how we see the very building blocks of life. To really get how this digital wizardry works, we first have to understand the physical law it's breaking. See, there's a very, very good reason why that image on the left is so blurry. It's not a flaw in the microscope, it's a fundamental barrier of physics itself. It's called the diffraction limit, and this idea goes way back to the late 19th century, figured out by scientists like Ernst Abbe and Lord Rayleigh. The principle is actually pretty simple. You just can't see something that's smaller than about half the wavelength of the light you're using to look at it. For visible light, that puts a hard stop at around 200 nanometers. Anything smaller or closer together than that, it all just blurs into one single blob. So why does this happen? Well, light, it doesn't travel in perfect laser-thin lines. When it passes through a lens, the waves actually spread out. They diffract. So even a perfect single point of light gets smeared out into this blurry spot. So when you have two tiny things right next to each other, their blurry spots just overlap and your microscope can't tell them apart anymore. That's the limit.
For decades, the only way to beat this limit was to invent entirely new, incredibly complex, and very expensive types of microscopes. But the breakthrough we're talking about today isn't made of glass or lasers. It's an algorithm. It's just code.
The solution is called MSSR, which is short for mean shift super resolution. And its core idea is kind of revolutionary. What if we could use the information that's already inside the blur itself to mathematically reconstruct a sharper image? It's a huge win for software over hardware, and it means this kind of super resolution imaging could become available to basically any lab. Okay, let's get into the weeds here because how MSSR actually works is just so clever. It's all based on this statistical idea called the mean shift. So imagine you're just looking at a cloud of data points. The mean shift concept is pretty straightforward. It calculates a vector, that arrow you see here, that always points from a starting point toward the densest area of the cloud. It's basically a mathematical way to find the center of a cluster. But here's the twist. For MSSR, the algorithm uses this idea in a brilliantly counterintuitive way. It's basically a four-step process. First, the algorithm looks at one of those blurry spots of light. Second, and this is the genius part, instead of pointing to the center, the math creates a map where the brightest part of the blur gets the lowest value. It creates a mathematical valley or dip. Third, it just flips that valley upside down. The lowest point instantly becomes the highest point of a new, super sharp peak. Finally, it just cleans that up, and boom, you've got a super resolved point of light. Okay, so that's the theory. But does it actually work on real-world images from a lab? Oh yeah, the answer is a resounding yes. The results are just fantastic. Okay, get this. The image on the left is of mouse chromosomes, and it was taken with an already advanced super-resolution technique. It's already way beyond the old diffraction limit. But now look what happens when you run that sharp image through MSSR. On the right, you can suddenly see that each chromosome is actually made of two distinct strands. Details that were hidden even to other fancy methods just pop right into view.
And the power of MSSR gets even crazier when it doesn't just analyze one picture, but a whole sequence of them from a short video. By pulling information from multiple frames over time, it can average out the noise and build an even more accurate picture. Here, it takes two blurry blobs and resolves them into two crisp points just 100 nanometers apart. Researchers have actually used this to resolve details as tiny as 40 nanometers.
And this isn't just for looking at test patterns. Here we're seeing real proteins inside a cell that's been infected with a rotavirus. The conventional image on the left just shows these fuzzy clouds of red and green, but after MSSR, on the right, you can see these intricate, almost ring-like structures. You can see how the viral proteins are actually organizing themselves. This is the kind of detail that helps scientists figure out exactly how a virus builds its little replication factories.
So, to put it all together, the transformation is just total. We go from indistinct, blurry shapes to sharply defined structures, from hidden details to a whole nanoscale organization being revealed. We're not just limited by the physics of our lenses anymore. We're pushing past those limits with pure computation. And maybe the best part? It can work with data from microscopes that labs already have, opening up access to this incredible level of detail for everyone.
So look, being able to see smaller things is obviously cool, but what does it actually mean for science? And, for us, why does hacking this physical limit matter so much?
Well, for one, it's already making huge waves. This isn't just some obscure algorithm stuck in a research paper. As this quote from researchers at Northwestern shows, major institutions are already incorporating MSSR into how they train scientists. It's becoming a standard part of the toolkit for modern biology. And the potential impact is just enormous. I mean, imagine applying these same principles to medical PET scans to help doctors spot the tiniest tumors way earlier than we can now. Or using it to visualize brain structures with a stunning new clarity to better understand neurological disorders. We could literally watch how a new drug interacts with a single cell, or, as we saw, map out a viral infection to build better and faster treatments.
For more than a century, the diffraction limit was a wall, an absolute dead end. Now, it's a hurdle we can just clear with code. And it forces us to ask this incredibly exciting question. Now that the invisible world is finally coming into focus, what unseen discoveries have been waiting for us just beyond the blur? What will we find next?